All right, welcome back. Another day, another dollar. Okay, next is DNA. This will be a talk about transcription and the regulation of transcription. This might take a while, but we'll try our best. And yep, let's start. First, transcription is the process by which an RNA molecule is synthesized by a DNA template. And the resulting RNA molecule serves as a copy of the genetic info encoded by or in the DNA. And so, why do we want to do transcription? Why? Okay, it's essential for protein synthesis, and because as the RNA is produced, it can be translated into proteins. And the basic steps of transcription would include initiation, elongation, and termination. And in both prokaryotic and eukaryotic cells, transcription involves the recruitment of RNA polymerase to the DNA. So you recruit RNA polymerase to the DNA template. All right. And you have prokaryotes. Transcription occurs in the cytoplasm due to the lack of membrane-bound organelles. And the process is usually simpler with fewer steps involved. So that's the big difference between prokaryotes and eukaryotes. 1 second, let me just check something really quickly. We have this there. Okay, we have initiation, elongation, and termination. I want to try and click down for this. Okay. Again, initiation, elongation, and termination. Initiation would be the first step in prokaryotes. And you have it as the important part is the -10 box, okay? And the -35 box TTGACA. That's recognized by the sigma factor of RNA polymerase. And the sigma factor helps the RNA polymerase locate the promoter, which would form the RNA polymerase hollow enzyme. So you have a lot of good terms here. Hollow enzyme, sigma factor, and etc. So the sigma factor helps the RNA polymerase locate the RNA polymerase hollow enzyme. In elongation, you have the RNA polymerase that is after it successfully locates the promoter. The RNA polymerase unwinds a small section of the DNA, which would create an open complex. And this unwinding provides the single-stranded binding part. So again, initiation in prokaryotic transcription, it is the -10, the -35, sigma factor, RNA polymerase, and RNA polymerase helps locate the promoter. Sigma factor, the sigma, helps the RNA polymerase locate the promoter, which would form the RNA polymerase hollow enzyme. And this hollow enzyme binds tightly to the promoter. Okay, next you have elongation. After initiation is done, the strand is the 3' to 5', right? And then it would synthesize it in the 5' to 3' direction. Termination, you have the row and intrinsic, very different, a lot of different sequences. And then you have in eukaryotic transcription. This is a bit more complicated. It's more complex due to the compartmentalization of the cell and the presence of different proteins or elements. It is in the nucleus. Transcription of eukaryotes it happens in the nucleus. RNA is produced and processed there before it is exported to the cytoplasm for translation. Okay. Prokaryotic versus eukaryotic transcription. You have initiation begins with the binding of the RNA polymerase 2. So RNA polymerase 2 binds. And the promoter, actually, it's the TATA box. TATA. TATA box. Is typically located around 30 base pairs upstream of the start site. And it also has like enhancers and silencers, but it also plays key roles in regulating gene expression. And the transcription factors such as TF2D, which binds to the TATA box using the TBP binding protein. It's a TATA binding protein. It's critical to form the pre-initiation complex. And then you have the other factors such as TF2A, B, D, E, F, and H would come in. Then you have elongation, and then you try to add a cap, and then you terminate it. We will go into that in a bit. So the key differences, some key differences between the prokaryotic and eukaryotic transcription is that in prokaryotes, the transcription occurs in the cytoplasm, while in eukaryotes, it is in the nucleus. So prokaryotes in the cytoplasm, eukaryotes, it is in the nucleus. So with the complexity of the machinery, how is it complex? Well, obviously prokaryote transcription is simpler with the RNA pol and sigma factor, but eukaryotic transcription has more things. RNA pol 1, 2, and 3, different transcription factors, silencers, enhancers, and etc. You also need to process it in eukaryotes, meanwhile not in the prokaryotes. And then for its factors, there's a lot more in the eukaryotes. And regulation is also more complex in the eukaryotic version. So just to start off with some questions, how can we explain the role of the C-terminal domain of RNA polymerase 2 in eukaryotic transcription? Well, the C-terminal domain of RNA polymerase 2, it is a long, repetitive sequence of serine and threonine residues, okay? Ooh, I forgot to include here. So earlier we finished the DNA topology, right? Meanwhile, replication is also 4, 9. Yeah. Okay, again, so what role of CTD, C-terminal domain? It's a long, repetitive sequence, serine and threonines in the tail, and it would coordinate various stages of transcription and RNA processing. And during transcription initiation, the CTD is not phosphorylated, no phosphate. And if you phosphorylate the CTD, at serine 5 using TF2H, that would mark the transition to elongation. So after that, RNA polymerase 2 moves into elongation, and CTD becomes further phosphorylated, particularly around the SER2 by CDK9, okay, of the positive transcription. And what's the difference between intrinsic and row-dependent termination of transcription in the prokaryotes? Well, intrinsic is when you have it's like a palindromic sequence in the RNA transcript which would form a hairpin loop, and that will lead to the dissociation of it. Meanwhile, in rho dependent termination, you need the rho protein. And what's the differences between gene expression and prokaryotes and eukaryotes? A lot. You have prokaryotes, you have the operators, like the operon level with activators and repressors. Meanwhile, eukaryotes, you have the RNA processing, chromatin, and spatial terminal regulation. So again, role of CTD, it's in the tail. If it gets phosphorylated, that would signal the movement from initiation to elongation. Differences between intrinsic and rho-dependent termination of transcription of eukaryotes. Hairpin loop versus they just needing the rho protein. Differences between regulation is that you have the activators and repressors. Meanwhile, eukaryotes, you have quite a bit of stuff. Okay? Because, wait, I have questions here. Yeah. Meanwhile, in prokaryotes, you have a bit of stuff. How does RNA splicing contribute to the diversity of protein products? That's a really good one. The RNA splicing is important because you have the spliceosome, right? And it consists of small nuclear ribonucleoproteins. U1, 2, 4, 5, 6. Let's not ask where 3 is. And we will talk about that more later. How do histone modifications and DNA methylation contribute to the epigenetic regulation of gene expression? It's a good one. Well, exactly how is it good? You have, if histone proteins are wrapped around, it's the proteins in which DNA is wrapped around. It's like a lumpia. And you form nucleosomes. So if DNA is wrapped around histones, that's a nucleosome. And modification to histones, if either you acetylate, methylate, phosphorylate, or ubiquitinate, can promote or repress transcription. So an example is you have histone acetylation. You acetylate the histones using HATs or histone acetyltransferases. That would, the HATs would neutralize the positive charge of histones, and that would lead to a more open chromatin structure. So again, histone acetyltransferases would neutralize the positive charge of histones, and that would lead to a more open structure. So HATs would the acetyls. If you acetylate, you neutralize the positive charge, the chromatin is more open, transcription factors can come in more, and RNA polymerase can access the DNA. Meanwhile, histone deacetylases would lead to a more compact chromatin structure. So, and that would inhibit transcription. Meanwhile, DNA methylation would mostly occur at cytosine residues, and that's within the CpG dinucleotides, and that would also lead to repression. So yeah, usually HAT earlier, that would neutralize the HATs would neutralize the positive charge, and that would lead to open and greater transcription. The HDACs, histone deacetylases, would lower transcription, and DNA methylation usually leads to repression also of gene expression. So that's why you have different things. Okay. And if you want, next is, so this is just the beginning of it. Neurons. Let's start with neurons. But this is a different topic. No, it's okay. Neurons have a highly complex and branched structure, and note that that has multiple dendrites and a long axon, and that's essential for their function of transmitting electrical signals. But since this is a different part, we can save that for later. For now, that is all. Thanks for watching. Bye.